Hello, my name is Monika Kuzma. I'm going to explain how to set up Q Box RP1LP Low Range Respiration Package. When you receive your Q Box, the tray with the analyzers will be inside. There will be a separate component, DCU solenoid, which needs to be connected. It will be delivered outside of the Q Box. Later on, it can be placed inside the Q Box. There will be two chambers, an insect chamber and an animal chamber, lizard, amphibians. These chambers can be provided in different lengths for custom purposes. There will be an accessory kit with some connectors, tubing, extra dry rite, which is used in a drying column. And there will be soda lime column, which is for scrubbing CO2 from gas during calibration of the CO2 analyzer. The first thing that you need to do is to check that everything is connected. I have everything turned on at the moment. I'll turn these off. But the first thing you want to do is make sure that everything is connected and powered. The power is coming from the Q Box to the main analyzer tray. Then we have data cables which carry the signals from the various sensors, from the flow monitor, CO2 analyzer, and there's two data cables from oxygen analyzer. One is for oxygen, one is for pressure. These cables are connected to data interfaces, which are these LabQuest mini interfaces. Each has 3 analog channels. They are labeled, so you know when you disconnect the sensors where to reconnect them. The DCU solenoid is connected to a Digi1 channel on the LabQuest Mini 2. And this DCU solenoid will control the flow of gas between reference and sample. So when everything is connected and ready, you can put the Q Box analyzer tray back inside, and you can power your analyzers and sensors. When you first power the CO2 analyzer, it needs to warm up, minimum 15 minutes, but half an hour is recommended. When the analyzer has warmed up, you will see the display showing actual CO2 values. When you receive your Q Box package, you may receive the analyzer tray separated from the Q Box for packaging purposes and shipment purposes. When that happens, I will show you here how to reconnect this analyzer tray with the Q Box. Inside the Q Box, there you will find a power connection, which should be connected right here, and you can screw this into place. Inside the Q Box, you will find all the data cables. They are already labeled, so you know which one goes with what sensor. QS151 is the CO2 analyzer. You would connect that, again, twist this to secure it. QS102 is the oxygen. You push it down, turn to secure. We have QS102P, which is the pressure sensor of the oxygen. Push it down, secure into place. And the final is the QG266, which is the flow monitor. Again, push it in, secure it in place. The tubing that goes in here, you cannot mess it up because there's two different fittings. One which is a push-in and one which is, this one you push in. You push them both in, but to release them, one you pull, the other one you push. So you can click them all into place, and they already have appropriate end on it, whether it's a female lure connector or a male lure connector, male with gas going out, female gas going in. So you click these into position. And now the Q Box is ready to go. The Q Box systems are modular systems, and you are able to take individual components from the Q Box and replace them with other components. So for example, in the RP1LP, we've got a gas pump P103, which is about a liter per minute gas pump. We have a 1 liter flow monitor, 2000 ppm CO2 analyzer, and a 25% oxygen sensor. However, if you're doing measurements on larger animals, more active animals, you can change the CO2 analyzer for higher range CO2 analyzer, 5% CO2 analyzer. You can change the flow monitor for 2 liters per minute flow monitor, and you can change the gas pump for a higher flow pump. To remove any of the components, you need a screwdriver, and there are 2 screws on the back of the sensor tray that has to be loosened up. You have to loosen both of these up. When you do that, each of these components can be taken out, removed, and replaced with another one. If there is any problems with any of the components, they're easily removable. You can send them back to Qubit for any type of service that may be required. Standard Q Box RP1LP consists of the pump, flow monitor, CO2 analyzer, and oxygen analyzer. However, if the user would also like to measure water vapor production from their samples, from their animals, then we can also add an RH temperature sensor, QS161, to the system. When this happens, we can combine, the user can replace the pump and the flow monitor with a single unit called flow monitor and pump, and that makes extra space for the RH sensor, which can be placed in this position. And in this setup, the Q Box RP1LP can be used for measuring both water vapor production, CO2 production, and oxygen consumption. So as I explained earlier, each of the components can be easily removed and replaced with another one. This would be a customized setup, and we can do any of that. While the CO2 analyzer is warming up, this is the time to open the software and connect it to the Q Box. The way we do this is we start by opening the setup file that was provided on the C404 disk, Q Box RP1LP setup file. You open this file before you connect the computer to the Q Box. Once the file is opened, there are 2 data cables that are provided with the Q Box. You can connect these cables to USB ports on the computer first. And then the other ends of the cables you connect to the USB ports 1 and 2 on the Q Box. It's very important that USB 1 is connected first. When USB 1 is connected first, you will notice in the software a sensor confirmation window will open, and there is a single star in the top menu. When you connect USB 2 next, a second star will appear, and that indicates that both LabQuest minis have been connected. Now this is the time where you select in the software which sensors are connected to what channels. The software does not automatically recognize that, except for the temperature probe, which here you can see stainless steel temperature probe is already listed under active sensors, and digital control unit, which is inside the DCU solenoid that I showed earlier, is already been automatically connected. But all the other sensors have to be connected manually. And just to review which channel they have to be connected to, you can look at the sensor connection window, which indicates channel 1 has the CO2 analyzer, channel 2 has the S102 pressure, channel 3 is the temperature probe, channel 1 of Mini 2 is the oxygen sensor, channel 2 is the flow monitor, and the Digi1 channel is the DCU. So these are selected here, and then you go across here and check that the correct channel is selected. So CO2 is channel 1 on Mini 1. You have to click connect, and then you will see the sensor show up under active sensors now. Now the pressure is on channel 2 of LabQuiz Mini 1. You click connect. That's been now selected. The oxygen is on channel 1 of Mini 2. Click connect. And the flow monitor is channel 2 of Mini 2. Once you've connected all the sensors, you will see that the readings from the sensors that are now displayed in these meters on the side, as well as in the top menu of the software. So now the software is ready to start collecting data. Initially, we're going to talk about calibrating the sensors and then data from your sample, insect or some other animal. When you were setting up your Q Box and connecting the two data interfaces, LabQuiz Mini 1 and 2, through these ports, it was very important to connect them in the correct order with USB 1 first and USB 2 second. This allows the software to connect the sensors correctly. But sometimes you may find that the sensors are not showing correct readings. There are two things that you need to check if that happens. For example, right now, the oxygen is a lot significantly lower than it should be when it's just regular air going through. So the first thing to check is to make sure that these minis are correctly, that they are activated, and you will see that through this green LED light. If the green LED light is not on, that means one of the mini is not connected. And if that happens, you need to just shut the computer down, unplug the two USB ports, and then go through the whole process of connecting from the beginning. The other thing to check when the readings are incorrect is that we have correct ranges selected. You will find that both the CO2 analyzer and the oxygen analyzer have two different range selectors. There's 2,000 parts per million, which is activated right now for CO2, and 500 parts per million. The two ranges require different calibration in that software, and the Q Box RP1LP is set up to be used with 2,000 parts per million range and with 25 kilopascal range for oxygen analyzer. So this is right now incorrectly selected. It's 100 kilopascal range selected instead of 25. So you need to change that. When you change that, you will see that the reading in the software is now correct, or close to being correct, 20-point-something% oxygen rather than 5, which is what the reading was before. So it's very important that the ranges are selected correctly, 2,000 parts per million for CO2, 25 kilopascal for oxygen.